Dear students, this is Dr. Pooja Thakkar, a biology expert. We are in the sixth session of Biomolecules, that is on enzymes. Enzymes are playing a very crucial role for the sustainment of our life. Okay, so first we need to undergo the detailed structural as well as the functional concept of the enzyme. Okay, each and every kind of metabolism in us involves a complex pathway of chemical reaction, as we have discussed in the last session. These enzymes are playing a very crucial role in that. They are the biological catalyst, which are specialized classes of proteins. So they are playing a very important role in the catalysis of the reaction to convert one molecule into the another. Okay, so they are basically helping to make the reaction super faster. But first, what we need to undergo is we need to understand the detailed structure of the enzyme. The overall enzyme is made up of, or it is called a holo enzyme. That holo enzyme is made up of two important ingredients, that is apoenzyme and cofactors. Now, what do you mean by apoenzyme? What I told, enzyme is a holo enzyme. Okay, and that holo enzyme is made up of two ingredients, that is apoenzyme and the cofactors. Now, apoenzymes are the proteinaceous part which are having the catalytical role, and these proteins are playing a very specific role on the different substrate, so they are providing substrate specificity. Okay, so what do you mean by cofactors then? So cofactors are the non-protein extra molecules. They get associated with the apoenzyme or the protein part, and they are helping in the transformation process to convert the substrate into product very quickly. Okay, so cofactors can be of varieties of types. So we need to understand the concept of cofactors now. The first kind of cofactor is the prosthetic group. They are the groups which are getting permanently associated with the enzyme, that is the apoenzyme. Okay, and they'll never ever release, and they are very mandatory for the enzyme to work. Okay, the next category is the activators, or they can also be called metal ions. So varieties of metal ions are necessary. They'll get bind with the active sites of the enzyme, then only the enzyme will get activated and it will be able to perform its task. For example, you can say zinc is necessary for the working of carboxypeptidase or carbonic anhydrase. So carbonic anhydrase is working, that is the enzyme which is converting H2O and CO2 into carbonic acid. Okay, so that is the reaction which is taking place by the presence of carbonic anhydrase. That enzyme need zinc ion, otherwise it will not be able to perform the task. Okay, so this is the importance of metal ions. And the third category of cofactor is coenzyme. The cofactor, or you can say the co-partner or the partner, which is not at all permanently bound with the enzyme, but they are attached with the enzyme for the action to perform, and then they'll get released. The best examples are vitamins. Okay, so few kinds of vitamins are attached to some kinds of enzymes, then the enzyme will be activated, it will perform its duty and its task, and then the vitamin will be released from that. Okay, so these are the varieties of cofactors, which are, as the name indicates, they are cofactors. They are the extra factors which are aiding the easy process of enzyme activity. Okay, one more note you need to mention here, as we have discussed regarding carbonic anhydrase, they are converting the CO2 and H2O into carbonic acid. That is the fastest enzyme till date known, which will convert 6 lakhs molecule of CO2 and H2O into carbonic acid, and vice versa. Carbonic acid can also get converted back into CO2 and H2O at the same time in every second. Okay, so this is the fastest enzyme. There are certain special enzymes. Okay, those enzymes are called ribozymes. The first category is ribozymes. As the name indicates, the RNA which is serving as enzyme or the nucleic acid which is serving as enzyme, that is ribozyme, which is the specialized enzyme. The other category is Metallo enzyme, the enzymes which contain metal ions, and they are working as the part of, or they are working as coenzymes. They are mandatory for the enzyme to work. Zymogens is the third category, or they can also be called proenzymes. They are the precursor of the enzyme. For example, if I'll say trypsinogen is the zymogen, it is in the inactivated version, but whenever the need come, that trypsinogen is getting converted into trypsin, which is the active form, and that will work on protein to degrade them into the smaller fragments. Okay, so the precursor molecule, or the primitive kind of inactivated enzyme, is called zymogen. The fourth category is allosteric enzyme. Now, these are the enzymes that are modulated by agents bind to them at a specific site other than active site. Now, here I'll make you understand the concept. What do you mean by active site? The enzyme is having the specific site where the substrate can bind. Then the enzyme will interact with that substrate. Then it will make the substrate to get, destructed. It will break the bonds of each and every substrate binded with that, and it will form the other kind of material that is called product. Okay, so active site is playing a very significant role for the product formation. Other than active site, there will be one more site which is called allosteric site, where some kind of activators or some kind of inhibitor can bind to activate the enzyme or to inhibit the enzyme. If the activator will bind, the enzyme will work more faster. If the inhibitor will bind, the enzyme will not be able to work. That's it. So those enzymes which are having both the sites together, active site as well as the allosteric site, they are called allosteric enzymes. Okay, now the next and the last category of special enzyme is isozymes. They are the different kinds of proteins which are having same enzyme activities. So two enzymes which are having same property, same task, same actions, they are called isozymes. Okay, now we have already learned about the structure of enzyme as well as the special categories of enzyme. Now what we need to understand is how these kinds of enzymes are basically working. What is the mechanism? A chemical reaction that takes place where the substrate is binded at the active site of the enzyme. The substrate bonds will be broken down. That kind of interaction of substrate and enzyme is called enzyme substrate complex. And then the enzyme accelerate the rate of reaction to break the substrate and to form the specific kind of molecule called product. Okay, thus enzymes are super selective. They are having catalytic role. They are playing the entire reaction, and these enzymes do not undergo any kind of permanent changes in them. That is the speciality of enzyme. No, not any kind of structural changes will happen in the enzyme. Only the structural format will be changed in case of substrate, and then the product will be formed. Okay, now to understand the mechanism, there will be two hypotheses that in the history few scientists have discussed different kinds of model, that how the particular substrate will get bind with the substrate, how the particular kind of enzyme is binding with the substrate to form the product. There are two kinds of hypothesis. One will be called lock and key model, and the other is called induced fit model. Now we are already knowing about the active site where the substrate can bind. Now in the lock and key concept, what we need to understand, the substrate has the specific active site specially designed in the same structure where it can bind to the enzyme, and then the substrate modification will take place to form the product. In between structure formed, which will be called enzyme substrate complex, and they are completely complementary to each other. Now in the other case, so this will be called the Fisher's model, or called the lock and key model, where the key and lock is specific, they are complementary to each other. Okay, but when the substrate is not at all having the same type of complementary binding site on the enzyme. Once it will get binded to the enzyme binding site will change. The active site will change as per the structure of the substrate, and then the enzyme substrate complex will take place. That is called induced fit model. Once the fitting has occurred, then the modification of the site will take place and then they'll become complementary. This has been given by Koshland. So this is called induced fit theory. None of the theory is perfectly accepted to understand the complex mechanism of the enzyme. Now we need to understand various factors which are affecting the enzyme activity. As we will be able to notice the diagram of pH, temperature, and the substrate over here, what we will be able to get the idea based on this is, as you go on increasing the pH level from 0 to neutral, the enzyme activity or the rate of reaction, the product formation will increase. That will reach the top. Okay, so at the acidic pH as well as at the basic pH, the enzyme will not be able to work properly. So the optimum pH for the enzyme to work is nearly neutral. Likewise, as you go on increasing the temperature, at initial stages you will be able to notice the rate of reaction is very less. But as you go on increasing the temperature, the rate of reaction will be super faster. So as you go on increasing the temperature, the rate will be faster and the more product will be formed. But after certain period, as you go on increasing the temperature, the enzyme will get denatured and the rate of reaction will be super down. It will not be increased, or it will not be the straight line. Okay, likewise in the case of substrate, as you go on increasing the substrate concentration with the enzyme concentration, both the numbers are nearly same or equal, then they'll be able to bind and they'll be able to form the ES complex for the product formation. But at certain time it will be saturated. So as you go on increasing the substrate concentration, but you will not be having the sufficient enzymes available with you, the substrate will remain as it is. It will not be able to get converted into product. The graph will be super steady at that time. Okay, so these are the different factors which will affect the enzyme activity. Now, to understand the enzyme activity in more detail, more precise, two diagrams are given in your textbook by which you will be able to know the rate of enzymatic reaction, as we have already discussed various factors. Now, if we consider each and every factor is easy, is comfortable, then when you are having the comfortable zone, each and every enzyme will work in the very beautiful manner. But this is the graph which is showing the difference. In the first case, in the blue line will indicate the enzyme's presence, whereas the red line is indicating the absence of enzyme. So what happens with the substrate in the presence and absence of enzyme, that has to be understood by this particular diagram. Each and every substrate has ability to get converted into product without enzyme. Okay, that is also possible, but it will take a very big time. So in the presence of that's why in the absence of any kind of enzyme, here it is mentioned no catalyst, that is no enzyme. So in the absence of enzyme, what you will find, the substrate is getting converted into the product, but the in between stage, which is called the transition state, is super bigger and more energy is required for that. That energy is called activation energy. The activation energy is needed to convert substrate into product. That energy requirement is super high, as well as the transition state will reach very later in this particular case where the enzyme is absent. But in the blue line, you will be able to notice in the presence of enzyme, immediately the substrate is using the activation energy and getting converted into product immediately by taking less time, by forming the transition state very quickly, by utilization of less activation energy, it will get converted into product. So enzyme catalyst will reduce the activation energy which is required, and thereby they will increase the rate of reaction, that is to convert substrate into product. The less time will be needed in the presence of enzyme. Okay, so this is all about this particular graph. In the second graph, what you will be able to notice that enzyme reaction with the substrate has been studied by many of the scientists, but the two scientists have done the research very interestingly and carefully. Those are Michaelis and Menton. So based on the overall research, they have given the formula, that is V is equal to Vmax into S upon Km plus S. Now in this particular diagram, or sorry, in the graph, what we need to understand is what is Vmax, Km and S. S in the bracket will be considered as substrate concentration. So in the x-axis, you will be able to notice the substrate concentration. As you go on increasing the substrate concentration, the rate of reaction will be increasing, as we have discussed earlier. This kind of graph we have already got in this particular case in the factors. Over here, the same graph has been represented in the next image, but we need to understand more terminology based on that. Okay, so now when we are focusing on the substrates, what we need to understand is as we go on increasing the substrate concentration, the rate of reaction will be increased, but at certain time period it will be super steady. When the super steady condition will come, at that time you need to draw the dotted line. When that dotted line is reaches to the y-axis, on that point you have to write the Vmax value, or that particular value has been noted as Vmax value. What is that Vmax? The maximum velocity. After that, the speed cannot be increased, as the graph also representing the steady condition. Okay, so at that time you will get the velocity or the rate of reaction as the maximum rate of reaction. Beyond that, the situation cannot be possible. So by chance, for example, if the Vmax value is 100, so what you need to predict the 50 value on the y-axis that has been shown as the second circle over here. Okay, so if Vmax is 100, the second dotted structure on the y-axis will be represented as 50. And at 50 circle, you have to draw the horizontal line towards the graph, where you will be able to notice one point where half of the Vmax has been mentioned. Okay, so that is true, right? 100 is your Vmax value. The half value of that, that is half Vmax, is on the graph, that is the 50 value, and that you are extrapolating on the graph by making the horizontal line, and then you are making the vertical line towards the x-axis, and that value will be considered as the Km value. Okay, so now what we need to understand is what is the Km value. Km is the constant value. Okay, so Km is basically represented as the substrate concentration, right? Because Km value you will here find on the x-axis, and the x-axis is representing the substrate concentration. So what you need to speak up is Km is your substrate concentration at which your Vmax is half of the maximum. Okay, so at 50 Vmax, whatever you are getting on your x-axis will be called as Km. Okay, so in technical terms, what we can say, Km is the substrate concentration at which the Vmax is half. So Km here is called as the Michaelis and Menten constant, and Vmax here will find by the help of the graph. So any of the value can be given to you to identify the other value based on this particular formula. and this is the standardized formula by Michaelis and Menton. Okay, so this is all about the basic information of enzyme. But one more important thing we need to understand here is the inhibitors. Now, what do you mean by inhibitors? Inhibitors are the kinds of molecule which are blocking or distorting or disturbing the active sites of the enzyme, which are mainly involved in the catalytic activity. So if these kinds of molecules are binded to the active site, the enzyme will not be able to work at that time. Okay, that is the very simple thing to understand. Now the inhibitors can be of two type. Irreversible inhibitor is the substance that is permanently inactivating the enzyme, which is not at all doing any reversible action by detaching with the enzyme, so it is very dangerous. But the reversible inhibitor, which is binding with the enzyme and that is getting detached from the enzyme after certain time period, then the enzyme will be back with its function. So that kind of inhibitors can be called reversible inhibitors. These kinds of reversible inhibitors can be of two types: competitive inhibitor and non-competitive inhibitor. As the name indicate, competitive is what? The inhibitor which is having the binding site which is similar to the substrate binding site. So in easier words, the active site where the substrate is binding, where the inhibitor is going to bind, so that the substrate will not be able to bind with that active site. So there will be a competition between the substrate and the inhibitor to bind at the specific one site. Whatever concentration, whatever molecule is having high concentration at that time, it will be able to bind at that particular location to block the enzyme activity. Okay, so enzyme and substrate are competitor here, then this kind of inhibitor is called competitive inhibitor. The next category is non-competitive inhibitor. These are the substances which are not at all having any similarity with the structure. They are not at all having any kind of structural resemblance. So the substrate and the inhibitors are completely different. So they are having the different binding site. As we have discussed earlier, that substrate is binding with the active site, and in this particular case, inhibitor is binding with the allosteric site or any other site. Okay, so they will little bit disturb the enzyme activity, but not at all disturbing the whole process. So the product formation will be continuing here, but that product can be of less concentration. The exact product will not be formed. Okay, so this is all about the inhibitors. And the last concept, that is nomenclature of the enzymes. Now what do you mean by nomenclature? The general name of enzyme is given by the name of the substrate to which it is binding. Okay, or the any other kind of activities it is doing. On that activity or the substrate, you will be able to add "ase" at the last. Suffix "ase" can be added. Okay, so whenever the enzyme is hydrolyzing urea, you can give the name urease. Okay, so based on the substrate, you will be able to notice the name. But in each and every case, it is not at all possible. So we have the list of category, that is oxidoreductases. So these are the enzymes which are doing oxidation reduction process. Then the examples are dehydrogenase and reductase. The next category is transferases. So they are transferring one molecule from one compound to the another compound. That is very easy to understand. Look, here in this case you will be able to understand the concept of oxidoreductases and transferases. A is the reduced compound and B is the oxidized compound. After the reaction, A will be oxidized and B will be reduced. The exchange will take place. Likewise, the transferases, A and B is attached with each other by forming bond. They are reacting with C molecule, and at the end of reaction, B is detached from A and that will bind with C. So it will be A plus CB. Okay, so based on the general scheme of reaction which is given here in the simplest version, you will be able to get the idea very easily. The next category is hydrolysis. Now as the names indicate, enzymes which are catalyzing the activity in the presence of hydrolytic cleavage. By water breakdown, they're getting energy. The best example is carboxylic ester hydrolases. So what they are doing, suppose A and B complex is there, they are reacting in the presence of H2O. Then H2O is split into two ions, that is H plus and OH minus. That H is binding with A and OH is binding with B. So those kinds of enzymes can be considered as hydrolases. The fourth category is liases. Liases are basically involved in the cleavage process. So those are cleaving different bonds called CC bond, CO bond, CN bond. The best example is decarboxylase. Okay, so liases are basically involved in the breakdown of that AB into A plus B. So they are segregating the two components together separately. The fifth category will be called isomerase. Isomerase is mainly involved in the isomerization process. Isomerization is what? The two formats of the same compound. The best example is D-glucose and L-glucose. Only structural orientation of few molecule is different. Otherwise, the structural formula is same. Both the glucose are having C6H12O6, but the arrangement of H and OH might be different. So they are converting one molecule into the another one with the same structural formula. So isomerase is what they are basically, if the compound is in that orientation A, B and C, so they are interchanging the orientation of the molecule, that is ACB. Okay, and the last category will be called ligases. They are joining the two different molecules together coupled with the hydrolysis process. Again here the hydrolysis will take place. The best example is synthase. So in the presence of ATP, A and B are joining together and ATP is getting converted into ADP and PI. Okay, so by understanding the structural format given in this particular table, you will definitely get the idea regarding various classes of enzymes, by which you will be able to get it in very easy manner. Okay, so this is all about the enzymes, how the particular structure of enzyme has been made, which are the special enzymes, what is the mechanism of the enzyme, which are hypotheses behind the mechanism, which factors are basically involved in the progress of enzyme as well as they are disturbing the enzyme action, how the particular enzyme is doing its own activity, what is Km and Vmax, which are the inhibitors, and what do you mean by nomenclature, how the namification of enzyme has been done. So this is all about enzymes here. If you have any queries, you can ask me in the comment box below. I know this is the very lengthy session of us, but this is the most important topic for the NEET examination as well as your 11th examination. Okay, to try to understand it very easily, see video 1 or 2 times or more times to understand the concept. Okay, and you can also call on the number called 990-980-4647 for more queries. Thank you so much.